let you introduce yourself and we can get with our last
Thank you very much. I'm very sorry for the rush, but it has been a crazy day and London is very far and the train schedule is terrible. So it's absolutely sorry for this. Thank you very much for being here at this, and I'm very proud to be here with you at your annual general meeting. My presence is the result of a long discussion that we started in last December with Ing and Mitesh at our Christmas event in UCL. My name is Lorenzo Perilli. I work in the neurogenetic lab of UCL, directed by Professor Henry Olden. And together with Ing and Adam, we discussed what about presenting you a pilot project proposal for this annual general meeting. We discussed about genetics and how difficult it is sometimes to obtain an answer. So that's why we designed this Rethinking HSP, a comprehensive genomic and clinical approach. So I will skip about me. I'm a pediatric neurologist. I graduated in Rome in Sapienza University and started my specialization in pediatric neonatology at the University of Siena, where I had the chance to encounter many patients with rare neurodevelopmental disorders. And I started my PhD in 2025 at the University of Genoa, but in a joint venture with Professor Holden at the UCL Neurogenetic Lab in Queen Square Institute. So we can absolutely skip on this overview that has been perfectly presented by Ye, but we just said that many HSP are monogenic progressive neurodegenerative disorders. What we mean with monogenic means that one gene can cause the disease if altered. So basically, the common symptoms that we all know are the spasticity of the lower limbs and the hyperreflexia that complicates the daily life of the patient. And this is the result, as said, of this length-dependent retrograde degeneration of these cables that connects the brain to the several districts of our body. It's a rare disorder, and many genes have been already associated, more than 100, but many others are still unknown and needs to be discovered. So our lab work in the field of gene discovery. So our interest with HSP starts in the early 80s with Professor Anita Harling that encoded the first clinical classification of HSP. And this is what we referred during all the other discussions. So we distinguish pure forms where the sole manifestation is the spasticity, whereas we have these plus forms of complex HSPs that present several other additional neurological features that we are going to discuss later on. And we have several inheritance patterns that have been can you hear me properly? That's okay, lovely. So we have different inheritance patterns that have been described for the transmission towards other generations. And this alteration in the genetics alters some key cellular processes that are involved in mainly in the maintenance and in the function of the neurons that rule the movement, as said before. So I know this is very basic and sometimes feel a bit too much didactical, but I wanted to prompt my presentation on a very basic terms just to be sure that everyone gets the same message. So basically, I want to describe you the DNA is a very big cooking book from a very, very efficient grandma that have put millions of annotation at the very edge of every page. And the DNA, the genes that are the recipe of this cooking book. And the cooking book, normally we use the recipe to make a cake, to make a first course. And this first course, this dessert or whatsoever, it's the final product of the expression of the gene, that it's the protein. So this work can be properly done, but sometimes some misspells arise over these recipes. So can alter the final product in many forms and have a deleterious or less effect on the final product. So, but how these genes, so-called, are passed on, and how these so-called misspells that we refer as variants are passed through generations. I'm just going to give you a few hints on the two main forms of inheritance, the autosomal dominant and autosomal recessive, that are the most recurrent among the genetic landscape of hereditary spastic paraplegias. But mainly, when you refer to an autosomal dominant condition, we just need one of the two copies of the genes altered. So normally when we have just one, normally genes have two copies in the DNA. Whenever just one is altered, it can produce a disease. Different situation is for the autosomal recessive conditions in which we need two copies in the same gene altered to produce a disease. So for the autosomal dominant, whenever you will undergo a genetic counseling, the risk, it will be said that it's 50%, but the reason why it's very clear, if you plot on this so-called Punnett square, you can see that the A, that is the deleterious affected allele, is present in on 4 children, just on 2. So these 2 patients will result affected, whereas for the recessive conditions, only a 25% will have both the unhealthy allele and will produce a phenotypic disease, whereas for 50% will be considered as carrier. So it's important, as said before, for genetic counseling. So this defects affects in HSP several machinery of this very precise several key cellular process of this very fine machinery. So we need to imagine like a neuron, like a very big city in which we need energy, we need fuel, we need transportation. And to do this, the mitochondria normally produce energy that is useful for all the mechanisms that maintain the activity and the oh, sorry. Yes. Sorry.
That's lovely. Thank you.
So the mitochondria normally affect, normally it's the fuel of this very high demanding city. And the products that are refined over the time and are constructed goes to the Golgi that's send it to this anterograde transport over this very big highway of microtubules toward other destinations in which other compartments, such as the endosomes and the reticulum endoplasmatic tubula, are active into the recycling of what is still needed and the disruption of what is not. So when these processes are altered, normally many proteins that are involved in these processes have been associated to HSPs because it disrupts the maintenance of the neurons that are very long and very demanding. So we defined some I will skip on this, but we defined clearly before what is complicated in the spectrum of HSP plus. As a pediatric neurologist, my focus is mainly on neurodevelopmental impact. So many patients with HSP plus several times present some cognitive impairment later on in life and some delays in the achievement of developmental milestones. And this can configure also the situation of global developmental delay. So when language and motor delay are present at the same time. But over the course of the disease, many other comorbidities can occur, such as the cerebellar ataxia, the peripheral neuropathies, and so many others that concord to give us this phenotype that we define as complex. So this is a very brief table that I wanted to give you, not because this is exhaustive. It's not. It's very small, and it's a very brief list of the main known SPGs. But the problem is that I wanted to show you this just to let you know that the dominant forms, as you can see, many times are more related to a pure phenotype, whereas the recessive that implicates that the two copy of the genes are affected. So the protein is severely disrupted, and it has a very deleterious effect on the production of this functioning protein, are usually with an early onset and more on the complex spectrum of the phenotype that we can find in HSP. So this should be longer as a list, but as said, many genes are still unknown, and sometimes the more we understand, the more questions arise. And it's a very tricky in the world of gene discovery, but it's also very exciting. But I want to start with a very interactive question, and I would like to raise your hand. How many of you have been genetically tested over time? So the majority. But the next question is a bit more tricky. How many of you know which genetic test have you underwent? So we see that probably not even a third of the room. And this is okay for me because I was expecting everyone to raise their hand and my presentation would have been ended. So I'm happy about this. So I can explain you what is the timeline of genetic testing that have been we went towards over the years in the UK. So normally we started and we used very much single genes assays to check whether one gene was responsible for the disease. But this was as reading one recipe from this very big cooking book. As we can expect in the clinical and genetic heterogeneity of HSP, this is pretty critical because we cannot base hope to obtain an answer only checking one specific gene, because it's like shooting to a very far target with a very, very tiny gun. So normally, so later on, we pass to agglomerate some genes and screen them in so-called panels that are still now used, and I will say you later how, but the gene panels are like looking at all the dessert recipes, but not looking at all the full manuscript from our grandma. So going later on, we started to use exome technique. Exome is a very, very important and still nowadays used technique that allows us to see the good DNA. So what encodes for real protein. And in here we had very, very important discoveries of new genes, and we still nowadays use to discover new ones. But it's like reading every recipe of the book, but missing some clue information that can be in the so-called junk DNA. So this so-called junk DNA is the DNA that normally is eliminated when we have the protein production. And this DNA, back in the days, we thought that it was completely useless. Now we are facing the problem that we don't know sufficiently information about these regions of the DNA that are very difficult to study. And normally now NHS provides as a gold standard for genomic testing genome sequencing. Nowadays, we're using for active line of research instead the long read sequencing. This is a highly, highly expensive technique, but can allow us to read every single annotation, every single details that we have in this very big cooking book. So how do we normally decide whether a variant, so this misspells, is deleterious or not? Normally, we rely into a very big spectrum of interpretation. So the genetic answer most of the time is an interpretation of what is the data, what are the data in the scientific literature up to date. So is not a binary answer of yes, no questions. It's a very demanding, time-consuming activity because we have some tools that help us, but most of the time we need to absolutely be sure to have correctly interpreted every result sufficiently. And the big problem of this interpretation, it's absolutely in the middle of the slide. So these so-called VUSs. The VUSs are variant of uncertain significance. These are negative results if we need to give a diagnostic and a clinically actionable result, and they are conflicting results that don't have any evidence to be sufficiently supported in their spectrum to decide it's pathogenic or it's more likely benign. It's in the middle, so we don't know and we need more proof. But this is what generates. This generates the need for reassessing these variants. Many times in every pipeline, in every country, this process sometimes is a bit tricky, and many VUSs historically remain VUSs, and not being reassessed sometimes remains the same, but many times can be upgraded to likely pathogenic, and so give us a diagnostic conclusive report. So how do we determine normally if a variant is pathogenic or not? We said that we use tools, but these tools are mainly in silico prediction. So it's like a computer-based analysis that provides us a percentage of how deleterious is the effect on the resulting protein. But we also use genomic data from population data from healthy individuals. So this big database that we normally refer to is NOMAD. It's composed of only healthy individuals, and the most immediate conclusion that you can understand is that if a variant that I'm looking at that I consider pathogenic in a patient is also present in a high frequency in the normal healthy population, it's very unlikely that this will generate a disease. Otherwise, the other patient would have been affected. And this is true not only for big population data, but also for clinical evidence collected from the same family, because many times informations in trio when we are working in a pediatric setting are present. So we have both the parents of the affected individuals, but many times it's tricky. So we need to collect more evidence into the same family to check whether is there any affected individuals. Later on, how we decide is really protein damaging, and it's really disease causing this variant. So we use several other techniques. So we use patient cells that can be derived from blood samples, but also from skin biopsies that I don't know if anyone had one, but it's a very, very non-invasive, minimally invasive technique that allows us to cultivate fibroblasts and other important cells that can let us understand at the very cellular level how the disease works. And also laboratory models that allows us to do this even without patients-derived tissues. When at the very last bottom of the functional studies, we have animal models that we use not only for looking at the very cellular level, but also at the very gross phenotypical level. What is the production of what is when I refer as phenotype, I refer as the symptoms that can be visible in the patient. So this helps us very much to deep phenotype the diseases. So whenever all these informations, all the population data, or the in silico tools are helping us to interpret the variant as pathogenic, this can be resulting in a conclusive diagnostic report. This, I'm going to skip to this because he explained it very well, but the most up-to-date studies are very, very early in the term that is not, there are not still already clinically actionable. The main one is on SPG50, some others are building up, but there isn't a precision medicine still in the HSP world, unfortunately. But what is the current genetic landscape of these diseases? And why I'm explaining this point to this audience? Because these are two of the most recent papers that have been published in literature, and one is still in press in a very important journal for us. And it tells us that in a very, very big cohort of Korean patients, the panel and the Sanger, that are two techniques very old, but also very important and basic techniques. The NGS panels is looking at just at the dessert recipes, so just a few genes, in this case 54, resulted in a diagnostic yield, so a diagnostic resolution of only 20%. Whereas for this less populated cohort of Greek patients with HSP, in the using exome sequencing, so an advanced molecular techniques, we obtained more than 60% of the of diagnostic resolution. So this leads us to the conclusion that advanced molecular testing can reveal more diagnosis and sometimes also can help us discovering new genes. So this led us to conceive a project that would involve your community, because we want to increase this diagnostic yield, because many times the genetic heterogeneity generates several problems in the analysis of the results. As for the problem that we discussed before with the VUSs, so sometimes are not reassessed. So we decided to build a project in which we can help and diagnose in the rescuing this situation and increase this diagnostic resolution. So to do this, we want to collect patients that have been that are not diagnosed, still lacking a diagnostic answer, and to make a diagnostic validation of the results that we will generate, but also reassess this historically VUS, and also identify further candidates for advanced techniques such as the long-read sequencing that I was mentioning you before. And also redefine the HSP classification, because the dichotomic classification of pure and plus sometimes is a bit too tight, and we see that there is a bit of a neurodevelopmental impact described on the patient, and many times in the neurology of the adult you can understand by yourself that it's a less touched ground. For a pediatric neurologist, sometimes when did you smile the first time, or when did you roll the crawl, it's a very important information, and sometimes this is very skipped in the adult neurology world. So We expect to stratify patients by prior genetic status. So we want to obtain this distinction in 3 groups. So the patients that have been already diagnosed that can work as a reference group for us for the phenotype. So all the symptoms and all the clinical history that we can take from this, and many of them probably will be affected by known genes, so SPG4 and so on. So we want to obtain, from these very brief questions I've made before, we will obtain a class of patients that haven't been diagnosed or it's negative or didn't underwent in their life under genetic testing. So why us? Why we should do this study? Thanks to the effort of Professor John Hardy and Professor Henry Olden, over the years, we accumulated more than 50,000 exomes that to give you a very rough impression, many centers feel that have a very big data power when they have from 1,000 to 10,000, but having 50,000 exomes to us gives us a very strong power in the interpretation of the results, because comes from a very underrepresented population that sometimes are lacking in this also population data from healthy individuals, but also from it's difficult to collect in this settings. So we have information that can help us to interpret it better, these variants. And this is just a few of our contribution in the scientific field of gene discovery. We characterized but also discovered several of these genes, and many of the known genes we discovered many times another form of transmission, genes that were historically autosomal dominant. Sometimes we obtained cases of patients with autosomal recessive transmission of the same gene. And currently, just to let you understand the proof of concept of how it works, I'm currently working on defining a new form of complicated hereditary spastic paraplegia caused by this biallelic, so 2 genes, 2 allele of the same gene altered. So it's autosomal recessive, as said before, forms of CHSP. And we started from digging in the data, so analyzing thousands and thousands of exomes, and finally obtaining that we found this patient that had a consistent phenotype among different cases. And this led us to help us in a clinical description of what is synergy-related disorder. And in animal models, we generated a zebrafish that can show us in an in vitro assay that the motility is reduced, so confirming the phenotype that we see on the patient. So we see it in the patient, but also in the functional model expressing, not expressing this synergy protein. And later on, the final stage would be also the pattern definition, if feasible, this so-called genotype-phenotype correlations. So whenever we have the symptoms, we try to see whether there is any link between any specific genetic alteration in this gene and some peculiar phenotypes in the course of patients. But it's a very, very difficult work and needs many, many cases to be absolutely certain. So this would be the pathway of this study. So we want to be sure that the diagnostic test with NHS is the first the first step of the study. So this can be done with a GP referral to our neurogenetics clinic in UCLH in Queen Square, and can be done in person, but also via telephone, and we can receive also samples posted. So or very kind nurse research, Ambreen can help us in this. And then we're going to screen these HSP genes in the genome. But what I said to you before about gene panels and why I said I will be saying something about these gene panels, because most of the time even NHS WGS focus on some, even if screens for all the genome, so also the junk region that we said before, many times use filter some panels of known genes. We are talking about more than 3,000 genes, so not a very limited panel. But most of the times, some regions can be underlooked. So that's why we step in as a research setting in this field. And after evaluating the patients in UCLH and performing WGS, we want in negative cases to reanalyze the data and going through advanced molecular techniques such as the long-read sequencing. sequencing. We have the lucky to have a very important long-read sequencing facility directly in UCL, and we have a very strong connection with the NHS Genomic Medicine. So we literally work at the very edge of diagnostic and clinical research. So what is deep phenotyping? Normally, when we refer as deep phenotyping, we said that phenotype is the sum of the symptoms that a patient presents. Deep phenotyping is going absolutely into this crucial definition, because many times patients see several doctors and it's very difficult to obtain a final answer, and mostly when we are in the world of movement disorder, because everyone sometimes see different things and it's very peculiar. So we want to analyze all the patients in general in UCLH and acquire all the information also regarding developmental milestones. How did you grow up over time, if available, this information obviously, but also screening the movement disorder through video assessment, because we have a very strong collaboration with several centers in the world for movement disorder, and we normally are very used to characterize them with the video assessment, and we collaborate also with these morphologists in case it's needed to check whether any peculiar shape of the face, of the hands, is present, and to better define these rare diseases. And we're going to see also the other investigations that have you made over time to have a very, very big clinical data of what you have done in your life. So normally what we expected to do is starting with a 6 months project in which we want to collect all the data over time with the patients that have been already diagnosed with HSP genetically confirmed. And we want to arrive to recruit patients that didn't underwent any genetic testing, to be obtaining the results in the next phase of the study, in the next 6 months, in which we will analyze actively the results and identify which patients can go in direction of advanced molecular techniques such as the long-read sequencing. So this will generate tons of results, but the key points will be that we want to increase this diagnostic resolution with a specialistic referral to a specialized neurogenetics clinic that work with HSP from the early 80s. And we want to reinterpret the available variants that have been stacked in this very long diagnostic journey, but also start to have a very trial-ready data set from which we can absolutely have strong information in any case for whichever project, and to go further into this broadening our scopes when we have preliminary results, because when you generate preliminary results, many times you can apply for bigger projects and involve more patients. And this absolutely is a translational framework, because we want to work, as said, at the edge with diagnostic assessment. So before concluding, I want to thank you very much for being here, our colleagues and our international collaborators from all over the world. And I don't know if Adam, you already said something. Early 2027, there's been a problem for the decided date, but we are now shaping up the final program, and all the details will be shared soon. But we want to invite you in UCL to have these open days in which you will have the chance to discuss with other clinicians and other researchers that are in the field of HSP, but also to provide your feedbacks on what needs to be done and what should have already been done. So thank you very much for your attention. I know it was such a lot of information, and I'm opening for questions. And if I don't have time, please take my email and write me. So I will be replying to everybody. Thank you very much.